So we will talk about the human genome sequencing and the variant analysis of a Pakistani individual. Before we start, I would like you to recall a slide which we have discussed before regarding the mutation in the DNA sequence, or the change in the DNA sequence, or variation in the DNA sequences. This is an important event during life processes, as we know that it is an important phenomena, that is change in the DNA sequences. And this is the major cause of diversity on this planet. We see different colors, different type of pupils, different attitude, different behavior, different diseases, different functions, because of the, at least partly because of the mutation, because of the change in the DNA sequences, because in the change in the DNA sequences. So mutation or variation in the DNA sequence is an important aspect. And as I mentioned before, that there are several types of mutation. There are several types of mutation. There can be several types of mutation. So the first and most common type of mutation is the point mutation, or which is also called as single nucleotide polymorphism, also called as single nucleotide variation. Single nucleotide variation. Point mutation, when one base pair is changed to another base pair in the sequence. Other type of mutations are called as indel. Either one or more base pair is deleted from the sequence, from the DNA, removed from the DNA, or one or more sequence base pairs, one or more base pairs are inserted in the sequence. So if it is, so this is called as indel, or deletion or insertion of the DNA sequences. Deletion or insertion in the DNA sequence. And there are other type of mutation called duplicate sequences, means when these sequences are duplicated. When the sequences are duplicated, that is a region of the DNA is duplicated. दो दफा आ जाता है, एक रीजन है डीएनए का, कुछ बेस पेयर हो सकते हैं और वो दोबारा रिपीट हो जाता है. This is called as, that is duplicate sequences. And inverted region भी हो सकते हैं, जिसमें that is the region, the sequence of the DNA is inverted, उलट जाता है, that is inverted in the sequence. जैसा होना चाहिए था, जिस sequence में होना चाहिए था, उससे वो that is inverted sequence में हो जाता है. This type of that is these are the major types of mutations in DNA, and there are some other types of, we will discuss later on. But basically, this basically I wanted to recall कि इस तरह की sequences mutation जो हैं वो DNA में होती हैं. ये mutation जो हैं, इनमें से कुछ mutations जो हैं वो they can cause diseases. Not all, very few out of very rare and few mutations can cause the disease. Okay, and then this is another slide which I have shown to you before. In this slide, we are mentioning over here that genome is only 0.1% different from other person. मतलब हमारा जीनोम जो है, मेरा जीनोम है, किसी एक इंडिविजुअल का जीनोम दूसरे इंडिविजुअल से how much is it is different, how much is it is different, that the genome sequence of one individual, that is genome sequence from one individual, this individual for example, how much it is different from another individual, whether it is male or female, doesn't matter, how much it is different. So normally it is 0.1% different from other person. 0.1% different. 0.1% of the 3 billion base pairs. 0.1% of 3 billion base pairs. When we calculated 0.1% of 3 billion base pairs, तो हमारे पास आई, it is approximately 3 million base pairs. 3 million base pairs in the genome, and is spread, that is in the all chromosomes all over the genome. Collectively, these difference is 0.1%. And this difference in the genomic sequences can be point mutation, one base pair mutation, single nucleotide variant mutation. This can be indel sequences, insertion or deletion mutation, or this can be duplicate mutation, inverted mutation, structural variation in which that is long stretch of DNA sequence longer than 1,000 base pairs is either deleted or inserted into the genome. So this slide is showing us a simpler aspect of the DNA variation. So I think this is and this slide again is a revision of the concept which we have discussed before. So having said that, how many okay, hopefully आपको आवाज थोड़ी सी अगर कोई इसमें है तो आ जाएगी इंशाल्लाह. So there is a lot of information, lot of a need for the human genome sequencing and human genome sequence analysis. And genome variation in human, as I mentioned, that with the exception of identical twins, all humans show significant variation in their genomic sequence. Okay, yeah, genomic sequence. It is therefore it is necessary that we should study the genome sequence variation in humans, in health as well as in disease, because it can provide information at genetic information regarding genetic diseases. It will give information about different oncogenes and mutations related to cancer. And it is also it is useful for the in the advancement in forensic sciences and so on. Okay, so the next slide. Human genome sequencing was initially, historically, you can say, is carried out by the Sanger DNA sequencing method. Sanger DNA sequencing. I'm sorry, अगर आवाज नहीं आ रही है, तो क्या सबको आवाज नहीं आ रही है? So historically, the genome sequencing has been carried out using the Sanger sequencing method, but recently means that more now it is around 10 years. During the last decade, recent advancement in the next generation DNA sequencing technology have greatly facilitated sequencing of complete human genome possible. These technologies, that is the next generation DNA sequencing technology, generate DNA sequences at extraordinary speed with reduced cost. Extraordinary speed with reduced cost, thereby enabling rapid scientific achievement and new application in biological sciences. Okay, so the NGS or next generation sequencing represents the I would like to mention over here that is the development in the sequencing technology, recent development in next generation DNA sequencing technology has resulted in the development of the genomic medicine science. So the genomic medicine has developed, as we know, today is mainly because of the development of the next generation DNA sequencing technology, as well as that is related bioinformatics development. So two sides. One side was the development of DNA sequencing technology by NGS, and on another side, the data analysis tools developed for the analysis of next generation sequences. The NGS, as mentioned over here, the NGS represent currently the only way to identify and evaluate rare and previously unidentified genetic variants at the moment, very quickly. The NGS has made it possible to develop a comprehensive catalog of genetic variation in human population, in human populations. The NGS of both universal and population-specific DNA variation The investigation of universal and population-specific variation will contribute to development of personalized medicine. So the NGS has a lot of application in the genomic medicine. Thank you. पानी ला दें पानी. ये ले लो. पानी ले. Investigation of both universal and population-specific variation. Hello? Is it? Hello? किसी माइक अगर खुला है तो प्लीज उसको बंद कर दें. जी प्लीज अगर किसी का माइक खुला हुआ है, ऑन है, तो प्लीज उसको स्विच ऑफ करें. Okay. Okay. So we were talking about the importance of we were talking about importance of the NGS, talking about importance of NGS, and we wanted using our information, using the information of the NGS, we wanted to carry out genome sequencing and the variant analysis of Pakistani individuals. Our motivation for the sequencing was because more than 90% of the sequences in the genome sequencing projects is from European origin. This is the Caucasian, Caucasian population. And for better understanding of genomic variants and its relationship to human biology, sequencing of additional representative genomes from a variety of geographical background is needed. Because at the moment, there is a bias. The genomic information is biased towards the European population, whether it is in North America or is sequencing done in the Europe. Complete genome sequences of Chinese, Japanese, Korean, and African and European has been reported, as well as a project called 1000 Genome Project carried out. Aim of the project is to provide deep characterization of human genome sequence variation as a foundation of investigating the relationship between genotype and phenotype. So in 1000 Genome Project, they tried to collect, they tried to collect DNA samples from all over the world. And however, the DNA sequence data from Pakistan, and in particular from that is the Indian subcontinent or the South Asian region, has so far unrepresented in genome sequencing studies and human genome variation studies.